All right, so we know about cells, which I believe is chapter 3 in your text. At the very least, it's the third lecture for this semester. At one point, I assumed it was chapter 3 in the textbook I was using. So the cell, smallest structural and functional unit. That means that none of the pieces inside the cell are alive by themselves. All of these ideas together is what we call cell theory. And cell theory is really sort of the basis of everything. The functions of everything that happens inside of you depend on the functions of cells. When we start talking about any system, we'll start by looking at the cells within that system because those cells determine how things work. In turn, what the cell is capable of is determined by the stuff inside it. We come back to this idea of structure and function complementing one another, the complementarity of structure and function. Even within cells, this is the case. The last part of cell theory is that cells come from other cells. And while that seems common sense, it wasn't always. There once was the idea that life just appeared. For instance, if you left meat out to decompose, the maggots would grow out of it. This was, of course, disproved by letting that happen under glass, where the flies couldn't get to it, because maggots are fly larvae. But if the flies don't land on the meat, there's no maggots, because cells come from other cells. Better than 200 different types of cells in people, differing in their size, their shape, their pieces inside, and what they do. Function is complemented by structure. Neurons may be very long, they're still microscopic. Typically, we consider the largest human cell to be the egg cell or the oocyte, which is about the size of a dot. Most cells you can't see with the naked eye. There's a reason for this. We have what's called the surface area to volume ratio. What that means is that as the size, in this case, of the cell increases, the volume increases at a much higher rate than the surface area or the area on the outside of the cell. Now, cells have to stay alive by taking things in from the outside through that outer surface area. So as the volume increases, the surface area doesn't increase as much, and you don't have enough surface area to support the volume they're in. There's a reason you don't see bacteria walking around, because it throws off that whole surface area to volume ratio. So cells are kept very small. The other reason for this is it makes cells expendable, honestly. Imagine if each of your muscles was just one big cell and that cell was damaged. Instead, there are billions of cells, and if you lose one, there's some backup there. They're expendable. So there's some different cells. There's red blood cells. Red blood cells are just sacks of hemoglobin. I mean, they don't even have a nucleus anymore. Fibroblasts and epithelial cells, those are all cells that are connecting things or lining things, whatever. There's muscle cells and fat cells and sperm cells. A sperm cell, the only cell with a tail or flagella on it. It's a nerve cell. Lots of different cells, different shapes, different functions. We're going to look at a generic cell. So while they all do differ, there's lots of things they have in common. And what we're going to be talking about is sort of the cell with all the pieces. So we have a plasma membrane or a cell membrane. That's the outside of the cell. The inside of the cell is called the cytoplasm. Most cells have a nucleus. Some of them don't, but most of them do. Some of them may have more than one nucleus. But here's our generic cell. You see the nucleus in the middle and all those different little thingies inside and the membrane on the outside. Now that membrane on the outside, the plasma membrane, is what we call a fluid mosaic. Fluid means it's constantly changing. Mosaic means it's made up of different things. So this fluid mosaic model is depicted as a phospholipid bilayer with lots of proteins dispersed throughout. Its function is to separate the inside from the outside, the intracellular fluid from the extracellular fluid. Intracellular, inside, extracellular, outside. Occasionally, you'll hear us use the term interstitial fluid. That's still outside the cells. It's just sort of in between cells. So here is our plasma membrane. So we have our, still not drawing. There you go. Expert at PowerPointing. So we have our phospholipids there. It's called a phospholipid bilayer because like they're back to back, like phospholipid. Phospholipid looks like this. We have phosphate head and these two fatty acid tails, and we have this bilayer structure like this. So they're back to back. Stuck in that phospholipid, we've got proteins. Sometimes you got proteins connected on, or carbohydrates connected on the outside. But there's lots of different molecules there, so it's a mosaic. And these molecules are variable. So it's fluid. Lots of lipids in the membrane. Phospholipids make up the majority of those. We have this amphipathic molecule with phosphate heads and fatty acid tails. Now, the phosphate head part of this is polar and hydrophilic. The fatty acid tails are nonpolar and hydrophobic. What that means, so there's our phospholipid layers here. We have hydrophobic middle, hydrophilic on either side. The structure is a little reminiscent of like an Oreo. So the cookie is hydrophilic, and the sugar lard in the middle is hydrophobic. Cell membrane. Now there are other lipids in the membrane. There's cholesterol that keeps it fluid. There's some glycolipids, which are lipids that have a sugar attached to the outside. There's lots of protein stuck in the membrane. And this really is super important because it determines how the cell is going to interact with all the stuff that's outside the cell. There's going to be enzymes here and transport proteins and receptors, lots and lots of stuff going on here with membrane proteins. And this is going to come up a lot throughout the semester. Some of them are loosely attached to the outside. They're called peripheral proteins. They may hook cells together. They may form part of the glycocalyx, which is involved in cell recognition. Here we see integral proteins that go all the way through, and then you see those little peripheral proteins that are kind of stuck to the outside. Here's a transport protein, which is going to let stuff in the cell or let stuff out of the cell. Some of these will be just channels, and some of them may use energy to actively pump something in or out of the cell. Here's a signal receptor. We may have a hormone, or for this semester, we'll have a neurotransmitter when a signal binds to its receptor. Now, these are proteins. Remember, your proteins have a tertiary structure. Sort of the rule we have here for proteins is anytime a protein binds to something, or anytime a protein stops binding to something, it will change the shape. It will change that tertiary structure. So in this case, we have that signal, whatever it is, it's a hormone or a neurotransmitter. It causes a change in the shape of the protein, and that's going to cause a chain reaction inside the cell. Or it may open a channel. It may cause that protein to change shape so that it's got a hole in the middle. Here is an attachment protein where we're attaching the inside of the cell to the stuff that's outside the cell to either hold it in place, or to guide movement, or to bind it to another cell. Here's an enzyme that carries out a chemical reaction in the cell membrane. Here's what's called a cell adhesion molecule, or CAM, and it's connected two cells together. Here's cell recognition. So that, for instance, cells of your immune system recognize your cells as yours. Those glycoproteins are like gang tattoos on your cells so that you can recognize your own cells. Now, cells are often held together to other cells. There are three types of membrane junctions that we care about: tight junctions, desmosomes, and gap junctions. A tight junction is just what it sounds like. Those cells are tightly held together to keep stuff from going in between them. Great example of a tight junction is at the blood-brain barrier, where the cells of the blood vessels are tightly held together so that stuff that's in your bloodstream does not leak out to the brain. Here is a desmosome. A desmosome is like a spot weld or a rivet in between the cells. It anchors them together so that tension applied to whatever this tissue is spread equally throughout. A great example of desmosome is in cardiac muscle, where those cells are held together so they pull on each other, and the tension of the heartbeat is spread throughout the heart. A gap junction is an electrical connection between the cell. This allows that electrical impulse to go from cell to cell to cell. The cardiac muscle is another great example of this. That electrical impulse travels through the heart in a wave because the cells are all electrically connected to one another. Now, one of the major functions here for our membrane is moving stuff in and out of the cell. The membrane is not a wall. It is selectively permeable, which means that some stuff can get through and some can't. There are different types of membrane transport, some that require energy and some that don't. A passive process means there's no energy required. In a passive process, we say something moves down the gradient or with the gradient. Down the gradient with the gradient, that means it moves from high to low concentration. Put a drop of food coloring in water. It's going to disperse throughout. It moves from the concentrated droplet and spreads out through the water. That's passive. Didn't require any energy. In active transport, energy is required. This happens in living cell membranes. We're pumping something against the gradient or up the gradient. We're going from low concentration to high concentration. So what determines whether something can actually just passively walk through a cell membrane? The first thing is lipid solubility. Things that are lipid soluble can freely cross the membrane. Things that are not, maybe. But if it's lipid soluble, it can go through. If not, there may be a channel there for it. And if there's a channel, then maybe it can go through. If all else fails, there may be a protein to carry it into the cell. So let's look at those things. There are different types of passive process or passive transport. The first type is simple diffusion. Things that are lipid soluble, nonpolar, hydrophobic molecules diffuse through the phospholipid bilayer. Here is a lipid soluble something going through the cell membrane. Steroids are going to freely cross the cell membrane. Anything that's a lipid freely crossing the cell membrane. Lipids freely cross the membrane. But obviously, your cell needs other crap. It doesn't just need lipids. So how do we get the other junk in? Well, we facilitate that diffusion. Facilitated diffusion means that we get some help. There's a protein that crosses the membrane that will allow something in or out of the cell. That's maybe a carrier protein or a channel protein or even both. The rules here, they are very specific. They don't just let anything in, they let one thing in. Also, they can be saturated, which means how quickly something moves in or out of the cell is determined by how many of those proteins are in the cell membrane. And you can regulate those. The cell will regulate how many proteins are in the membrane and what they're doing. So let's look at a carrier protein first. A carrier protein, remember when a protein binds to something, it changes shape. So here you have something, glucose on the outside of the cell. We want it inside the cell. It binds to that protein. The protein changes shape. Now it's inside. No energy used. We're still going from high to low concentration. This is still diffusion. It's just using that protein to get that hydrophilic molecule inside the cell. We also have channel proteins. Channel proteins are just what they sound like. They're channels. So here you have whatever that is. It's hydrophilic going through that channel. Now, like I said, these are specific for different things. So when you think about this, these channels or even carriers, they're very specific. If you have kids, when they were babies, somebody gave them that stupid shaped ball. You know, the ball, it's got the different shaped holes in it and the little blocks that go through the different shaped holes. You're never going to keep up with all those freaking things. You're going to get like 40 of them. Oh, look, that one's got different shapes. Oh, look, that one's pink or whatever. I don't know. I don't have anything that's pink. Who am I kidding? But, you know, the circle goes in the circle hole, the star goes in the star hole, square goes in the square hole. Same principle here. Circle goes in the circle hole. Sodium goes through the sodium channel. The potassium-shaped molecule won't fit through the sodium channel, is what I'm getting at. So they're very specific. Osmosis is the diffusion of water. Now, water will go through the membrane, and it goes through channels that are called aquaporins. But water is the solvent. Everything's dissolved in water. So how does diffusion work there? Well, here you see those little Mickey Mouse shaped water molecules going through the lipid bilayer and through that aquaporin. Well, here's the thing, is water concentration is determined by solute concentration. If you dissolve something in water, that something displaces the molecules of water. Osmolarity is the measure of the total concentration of the stuff that's dissolved in there. If we separate solutions of different concentration, we get osmosis. Now, here's your rule for osmosis. Water follows stuff around. Technically, water follows around the osmole, but water follows stuff around. Water follows dissolved stuff around. How we would look at this experimentally. So in this situation, we've got this weird U-shaped tube, which I assume totally exists. And it's got this little membrane right there inside. Now, you can see this membrane, it's got a pretty big opening here where those little red dots can go through that little opening. Now, if you do this and you put, like in this case, it says sugar, if you put it all on one side, you're going to get diffusion. The molecules move down the gradient until you get equilibrium here, because those little red dots can move through that membrane. But what if we change that membrane so that the little red dots can't fit through it? Like so. Now those dots can't get through here. So what happens is, because the little red dots can't move, the water moves. the water will move through and it goes this way. That is osmosis. Water follows stuff around. Real life example. Still too cold outside, but when it starts getting a little warmer, the snails will come out. You can take a snail for science. I have to say that. Put salt on it for science. What happens to the snail? For science. Well, what happens is snails don't have skin like we do. The surface of the snail's body is a semi-permeable membrane. So when you put salt on it, water follows stuff around. Water goes to the area of higher concentration. So the water rushes out of all that snail's cells to meet the salt that's outside it now. Of course, killing the snail for science. That's osmosis. When osmosis happens in your cells, water is going to enter or leave the cell. Now, this will screw stuff up. You don't really want this to happen. Tonicity is the ability of a solution to cause a cell to either shrink or swell. Your cells are bathed in an isotonic solution. Isotonic means that we have the same concentration of particles on the inside as we do on the outside. So there's not a net gain or loss of water. In a hypertonic solution, you place the cell in something that has a higher concentration on the outside than we do on the inside. And in a hypotonic solution, we have a higher concentration on the inside than we do on the outside. What does that look like for your cells? Like this. In an isotonic solution, you see that red blood cell. It looks like a red blood cell. Water is moving in at the same rate it's moving out. Put that red blood cell in salt water, for instance, or something like seawater or whatever. Well, now there are more dissolved particles outside the cell than there are inside the cell, so the water goes out. In a hypertonic solution, water will leave the cell. In a hypotonic solution, we get the opposite situation. Now, there's nothing outside the cell. There's still stuff inside the cell, so water comes in. The cells take on water, and they will eventually explode. So in a hypotonic solution, the cell swells up. So here are our passive processes. Diffusion, facilitated diffusion, which is it's the same as diffusion, but with a protein facilitating that movement. And then osmosis, which is the diffusion of water. Active transport requires energy. And there are two types of active transport, active and vesicular. Now, both of them are requiring energy, and remember, energy is ATP. Active transport moves something against a gradient from low to high concentration. And it's going to require a carrier protein that uses ATP. Now, there are two types of active transport. The first type we'll look at is called primary active transport. We directly use the energy of ATP to change the shape of the protein, and we pump something across the membrane. Most important here is our sodium-potassium ATPase pump. The sodium-potassium ATPase pump is located in every plasma membrane of the cells in your body. It is involved in primary and secondary active transport of everything. It is extremely important because it also maintains the electrochemical gradient necessary for your nerves and muscles to work. It maintains the water balance of the cells. Extremely important protein, the sodium-potassium ATPase pump. Let's look at that. So here we have sodium on the inside of the cell, and it binds to that pump. Then ATP donates a phosphate and the energy therein, which changes the shape of the protein and throws that sodium out. Potassium binds to the protein, changes the shape again, and now it's pulled back in. Every time this works, we get 3 sodium out, 2 potassium in. 3 sodium out, 2 potassium in. That's important. This is going to be involved in what's called the electrochemical gradient later. Secondary active transport depends on that gradient that your primary active transport just made. So we pumped all that sodium out, so now there's a lot of sodium outside the cell. It would really like to come back in. And we're going to use that to push something else. This is co-transport. Now, what we'll look at is called a symport, where we're moving two things in the same direction. But there are antiports where they're moving in opposite directions. Here is our sodium glucose symport. We pumped all that sodium out. It really wants to come back in, and we'll let it back in with a glucose. So as that sodium comes back in through the membrane protein, it pulls glucose with it. Glucose doesn't particularly want to go in, but it gets dragged along for the ride with sodium. So this is secondary active transport. Notice there's no ATP being used here. That ATP was used here. So it still requires energy, just not directly. Vesicular transport is moving big stuff. It's using a vesicle, this encapsulated stuff across the membrane. And obviously this uses ATP as well. This can be exocytosis, which means moving something out of the cell. Endocytosis in the cell. Transcytosis is in one side, out the other. Or even within the cell where we're moving vesicles around. Where these vesicles will be coated in proteins. Sometimes we have very selective endocytosis and transcytosis. It's called receptor-mediated endocytosis. Really, what you should know about endocytosis at the moment is that its active transport involves this vesicle. So there's our receptor-mediated endocytosis. So very specific, and you can see that being moved into the cell. Phagocytosis is another form of endocytosis. Phagocytosis means cell eating. So here you see the cell being fed hamburger meat or whatever they're feeding cells these days, and it's wrapped around it and bringing it in. Fluid phase endocytosis is called pinocytosis. It means cell drinking. And so here it's just gulping fluid outside. This is not specific. Receptor-mediated endocytosis, again, this is very specific, where things bind to receptors on the cell membrane and then they're all brought in together, like so. Anything that binds to a receptor is called a ligand. So when you see that term ligand, that's anything that binds to a receptor. Exocytosis is the opposite. We're throwing something out. So here you see cell junk, whatever, surrounded by that vesicle, and that vesicle goes and binds to the membrane, opens up, and throws it out. All of these things are active transport. They require ATP. Now, going back to this idea of the sodium-potassium pump, we pumped 3 sodium out, we pumped 2 potassium back in. That means we pumped more positive charges out than we pumped back in. This creates potential. It means that the outside of the cell is more positive than the inside of the cell. Potential is the difference in voltage. The resting membrane potential is the voltage measured in the resting state of cells, usually somewhere between -50 and -100 millivolts. For our purposes, we're usually saying it's like -70-ish millivolts, sometimes -90, but right now like -70 millivolts. So, right, we pumped all those sodiums out, potassium back in. Potassium leaks out very quickly, and we're left with a negative charge on the inside. This will be important. That separation in charge can be used to harness that electrical potential. More on that when we get to nerves and muscles. Now, cells obviously interact with stuff outside the cells. We've got this glycocalyx, these carbohydrates attached to proteins and receptors, cell adhesion molecules where cells are stuck to each other, membrane receptors we're going to spend a lot of time with, especially in terms of chemical signaling where we have a ligand and a receptor. For our purposes this semester, neurotransmitters are the ligands we'll be talking about. Sometimes that receptor triggers a chain reaction. We have what's called a G protein link receptor, where the neurotransmitter binding activates this cascade effect within the cell, like so. And you can see that ligand binds to its receptor and sets off this giant chain reaction. Everything inside the membrane is the cytosol. And, the cytosols that use the cytoplasm, so all this crap inside the cell, lots of organelles, and some other stuff. Now, here, we're going to be very general. The organelles, what I care about you knowing here is really function. I don't care about how that works. For example, you need to know that mitochondria make ATP. At the moment, you don't need to know the process of oxidative phosphorylation. That's a topic for another semester. It's good enough that you know that mitochondria make ATP. We'll talk about details later. Some organelles are bound by membranes. The mitochondria, peroxisomes, you see that list there of membranous organelles, non-membranous organelles, the cytoskeleton and centrioles, and ribosomes. They don't have a membrane. The mitochondria is kind of cool because it has its own DNA. It reproduces independently of the cell. And this is where ATP is made. Mitochondria make ATP. They look like that. Again, I'm not worried about how. Don't worry about glycolysis and the Krebs cycle and electron transport right now. Mitochondria make ATP. Ribosomes are a protein in a molecule of ribosomal RNA, rRNA. Ribosomes make proteins. You should know that ribosomes make proteins. Some of them are just floating around. They make proteins that stay inside the cell. Some of them are bound to the rough endoplasmic reticulum, and they're going to get exported. The proteins are going to be exported outside the cell. The endoplasmic reticulum, a system of tubes and membranes and stuff, and there's two types. There's a rough ER and a smooth ER. And you can see that here. The rough ER is called that because it's all those ribosomes stuck on it. The smooth ER is not studded with ribosomes. The rough ER, ribosomes make proteins, so the rough ER is involved in manufacturing proteins. You can think of the ribosomes as like small children in a sweatshop making the shoes you're wearing, and the rough endoplasmic reticulum is the factory where your tennis shoes are made. Those tennis shoes are going to be exported. More on that in a bit. The smooth ER, there's no ribosomes there, so it's not involved in protein manufacture. It's doing things like detoxification or maybe storage of calcium in muscles, maybe making steroids. There's some different functions for the smooth ER, but it's not involved in protein manufacture. The Golgi apparatus is like FedEx in your cells. So once the ribosomes are done making the proteins for the rough endoplasmic reticulum, the rough endoplasmic reticulum sends them to the Golgi apparatus, which then ships the shoes outside the cell. Shoes, proteins. Got lost in my own analogy. So here we see this. You see the rough endoplasmic reticulum, again, sweatshop, tiny little ribosome children there making proteins. And then that gets shuffled off to the shipping center, the Golgi apparatus, which then decides where to ship it. Lysosomes are digestive organelles inside the cell. There's these enzymes that are going to break stuff down. Now, if we go back and look at this, you see these little vesicles come off and then they join the next organelle. And then you see the little vesicle here and it joins the cell membrane, or it joins the lysosome over here. They're all sharing membranes. They're part of what's called the Endomembrane System. This system of organelles working together to produce and export stuff. The endoplasmic reticulum, the Golgi apparatus, the vesicles in between them, lysosomes, and the nucleus, the membrane around the nucleus, are all part of the endomembrane system, sharing the membrane. Here's another view of that, and you can see that membrane being swapped in between those organelles. Peroxisomes are sacs of other things with other enzymes to help detoxify things and break down things called free radicals. The cytoskeleton we don't usually see in most of these diagrams because the cytoskeleton is this sort of system of filaments throughout the cytosol where everything is. Microtubules, microfilaments, and intermediate filaments. Microfilaments are made of protein called actin. They're involved in the shape of the cell and movement and moving stuff in and out of the cell. Here's microfilaments forming this network around the nucleus in the cell. Intermediate filaments are like ropes, and they'll attach to desmosomes, and they resist forces that are pulling on the cell. These intermediate filaments. Microtubules are these hollow tubes that are made by another organelle, and they determine shape and distribution of organelles. The microtubules make sort of a highway inside the cell. When stuff inside the cell moves around, microtubules are involved. Motor molecules are like little taxis that jump on this microtubule highway, powered by ATP, moving stuff around. So here you see this vesicle, this little motor molecule walking along the microtubule. There's some really fun animations of this, and you see the stupid little motor molecule walking along that microtubule. The centrosome makes microtubules. It's kind of near the nucleus. It looks like this. It looks like it belongs on the side of a religious candle. I don't know why it's always drawn like this, like it's got like a halo around it. Weird religious candle organelle there. But that's the centrosome making microtubules. There are extensions of some cells. We have cilia, we have flagella. These move. So they're microtubules and motor molecules. Cilia will move something across the top or the surface of a cell. There's only one cell in the body that has a flagella. That's the sperm cell, and it moves the cell around. Here's the organization of either the cilia or the flagella and the arrangement of those microtubules. Here you see the movement of cilia moving mucus usually along the surface of the cells. Other extensions include microvilli. Now, where cilia and flagella move, microvilli aren't there for movement. Microvilli are extensions of the plasma membrane to increase the surface area of the cell. Anytime you see microvilli, you need to associate that with surface area, and that surface area with absorption. The more surface area you have, the more you can absorb. So cells that are specialized for absorption are going to have microvilli. Here we see microvilli. The nucleus, where all the genetic materials maintain. DNA, right? RNA. Blueprints for everything. Most cells have at least one nucleus. Red blood cells don't. Skeletal muscle cells, some cells that dissolve bone, and some of the liver cells, even some heart muscle cells might have more than one nucleus. There's the nucleus. In the center, you see that darker body, the nucleolus, attached to the rough endoplasmic reticulum on the outside. The nuclear envelope surrounds the outside, double membrane barrier, rough endoplasmic reticulum is continuous. Here's a picture of the nuclear envelope. You can see the little pores there and this sort of lamina that holds the nucleus together. The nucleolus in the middle has ribosomal RNA. It's involved in assembling ribosomes. Inside the nucleus, we have DNA. Strands of DNA called chromatin, trapped on these little proteins called histones, and there's some RNA as well. And this is arranged in things called nucleosomes. Now, when the cell gets ready to divide, this chromatin is going to condense into chromosomes. Like so. So there's DNA, there's chromatin, and then you can see it condensing down into that chromosome at the bottom. The cell cycle, changes that happen in the cell from the time it's formed to the time it reproduces. Two big parts, interphase and mitosis. Interphase, from when it forms to when it gets ready to divide. Inside the nucleus, DNA is floating around this chromatin. And there are four big phases for interphase. Gap 1, where the cell's doing its cell thing. It's growing, it's doing its thing. The S phase, it makes a copy of DNA. And the gap 2 phase, it gets ready to divide, or mitosis. Now, some cells have a gap zero phase. A gap zero cell is a cell that has stopped dividing entirely. It's amitotic. It's not going to divide again. So here, in the green here, we see interphase. There's our gap 1, S phase, gap 2 phase. In the yellow, you've got mitosis. So here's interphase. Interphase is what we're normally looking at when we look at these pictures of the cell. Now, during the S phase, DNA is going to make a copy of itself. That alpha double helix is going to unwind itself. An enzyme called helicase untwist it. And then we make a copy of DNA. Each side of that double helix serves as a template for the new one. This is called semi-conservative replication. It looks like this. Now, I'm not worried about your 3' to 5' end and your leading and lagging strand. That's not important. This happens during the S phase. We'll make a copy of the DNA. Then, once we've made a copy of the DNA, we can do mitosis. Now, mitosis is how you grow. It's how you repair. Mitosis gives you identical copies of the cell. We don't see mitosis in neurons or skeletal muscle cells or cardiac muscle cells. Those cells are in the gap zero phase. So two big events here, mitosis, which the nucleus divides, and then the cytokinesis, where the cell just splits in half. 4 phases for mitosis: prophase, metaphase, anaphase, telophase. By no means am I going to ask you to identify these on a slide. This is how cells replicate. So here's prophase, right? We condense in the chromosomes, the nucleus starts to fall apart. You get this mitotic spindle forming here. Later in prophase, the nucleus dissolves and the little spindle attaches to the chromosomes. In metaphase, they line up all in the middle. And then in anaphase, we pull them apart. Now they're going to get pulled back this way, and they're going to pull back this way until we get to telophase. And in telophase, we get a new nucleus on either side, and nucleus forms, and the cell splits in half. The important part for you to understand about mitosis is that mitosis gives you 2 identical copies. This is growth. This is repair. This is not reproduction in terms of like producing offspring. That's different. The cells here are identical cells. Now, how does the cell know when it's time to grow or when it's time to divide? Well, if it gets too big, it's time to divide. And then there are different chemical signals that will tell it it's time to divide, hormones and these growth factors and other things. Just as important is when's it time to stop. If they start running into each other, it's time to stop. There are also things that will stop cell division. We have genes called repressor genes that will stop that. Now, DNA is the blueprint for making proteins. A gene is a segment of DNA with a blueprint for one protein or polypeptide, and every 3 letters on that DNA forms our genetic library. Every 3 letters codes for a specific amino acid. Here is the important thing for this. DNA, RNA, protein, transcription, translation. Transcription is taking DNA and transcribing it as messenger RNA. Back to my sweatshop analogy. So your ribosomes, the little kids working in your sweatshop, they can't read. You don't want them to be able to read. If they could read, they might like form a union or something. You don't want this. They can't read. The instructions for making your Nikes or other shoes, I assume, are written on DNA. That information is transcribed as a messenger RNA. So you draw them a picture or whatever. And then that is taken out to the ribosomes, and then they follow that instruction on the messenger RNA, which is put in a form that they can understand. And they translate that into a protein. Transcription, translation. DNA, RNA, protein. Messenger RNA is the RNA that carries the instructions for the protein from the DNA to the ribosome. Now, there are other types of RNA. There's ribosomal RNA, which is actually part of the ribosome, and transfer RNA, which is going to bring in amino acids for this process of translation. Transcription is taking the code on DNA and transcribing it as messenger RNA. I'm not worried about like RNA polymerase or how RNA knows where it is. That's what transcription looks like there. We transcribe the message that's written on DNA as messenger RNA. Translation is then taking that code that's on the messenger RNA and turning it into the sequence of amino acids in a protein. Every 3 letters on DNA is represented by a codon on the messenger RNA. Here's the genetic code. Now, one thing to notice here is how redundant it is. There's like all these 6 things that code for the same amino acid. What that means for you is that if something's screwed up here, you're going to be fine. Any of these 6 things will code for the same amino acid. So if there's a mutation somewhere and one of those U's becomes a C, you're probably still going to get the same amino acid. Now, what we get from this then is translation, and we get the primary structure of the protein. We bring these amino acids in and we connect them together. Here's translation. So there's our primary structure coming off here. And of course, then we're going to get secondary structure and tertiary structure. All right, so you're made up of a lot of cells, but you're also made up of other stuff too. There's other stuff outside the cells, fluid and secretions and the extracellular matrix, stuff we're going to talk about next when we get to tissues. So this will wrap things up for cells. Really quick overview of cells. Next, we're going to start talking about tissues. And we're moving right along. So after tissues will be skin and then bones. So I will see you there.